Hi, this is Nick Cap, and this is your 15-minute wrap up of the lecture that I gave on Wednesday, the 26th of January. I'm going to share my screen here for a little bit. Let's see if I pop up with that. And so basically what I'm sharing with you here on the screen is the page on Canvas. We're looking at week 2. Unit 4, Mechanisms of Evolution. And I did want to go through chapters 24 and 25. And in my previous 15 minutes, I went through chapters 22 and 23. We will be having an exam on the 2nd of February. And so there's a review list here that you could go on and take a look at that. There are some materials that you can still do here. That is the the flip grid and the observations one. So those materials are still available, and you can kind of hand those in by midnight tonight, which is, I believe, the 27th. So good luck with that. So what I wanted to do is real quickly kind of go on to chapter number 4, which is, again, looking at the origin of species, chapter number 24, which is looking at the origin of species. And I am going to pop on this one, and I'm going to set up a slideshow. Now, I pulled a subset of slides from this in order for us to take a look at this. But basically, what we're looking at in evolution is this process that we call speciation. And this is the process by which one species splits into 2 or more species. And again, it's the focal point of evolutionary theory, right? Because what you do is you go on and you have that slow adaptive change. Now, in looking at speciation, we also need to look at what we call microevolution. These are the changes in the allele frequencies in the population over time. And ultimately, these will lead to speciation. If 2 species are too different, then they won't be able to mate. And we'll talk about there's a number of reasons that would kind of prevent an organism from mating, and some of it's biochemical, some of it's behavioral, some of it's due to space, etc. This speciation and microevolution are important, but another thing that we want to talk about is what we call macroevolution. These are the broad general patterns of evolutionary change. And with macroevolution, we're going to have a bunch of little speciations. We're going to have new species occurring. We're going to have species kind of dying out. And basically what's going on is when we talk about macroevolution, we're talking about these large changes that allow groups of populations to kind of go into new areas. And so again, it says here, speciation forms a conceptual bridge between macroevolution and macroevolution. Species, for the Latin word meaning kind or appearance. And so a lot of times, especially when we're looking at fossils, you're just looking at the shapes of bone and you're trying to determine a species from that, which can be difficult sometimes. Again, in modern biology, we're going to be looking at morphology, the structure, physiology, the biochemistry, and DNA sequences in which to group organisms. So it's not just necessarily looking at kind of what they look like. The biological species concept states that a species is a group of populations whose members have the potential to interbreed in nature and produce viable and fertile offspring. And so again, in this biological species concept, you have to have 2 species come and mate together. What again, we're finding cases of this where basically an organism doesn't necessarily have sex that we think about. And again, the bacteria, that's been a big problem for the bacteria, where this kind of doesn't happen at all. So what is that species? Now, in order to get a species, a lot of times you need what's called a reproductive barrier, and that a species needs to go on and evolve by itself for a while. And so these reproductive barriers, there's a number of them that we have. The first one is habitat isolation. So if you have 2 organisms that won't live next to one another, they're not going to be able to mate because they're physically not going to be able to go there. In temporal isolation, you have species that are separated by time. If one is active during the day and sleeps at night, and one is active at night and sleeps during the day, there's no time that they get together in order to do that. And then behavioral observation, if you look at the slide, you have the blue-footed booby, which does this little dance and shows off its blue feet. And again, if you can't do the dance, then you're not going to mate. And so again, those are what we call prezygotic barriers. So these are going to be happens, physically you're not mating. In mechanical isolation, we have where an organism, again, may have a penis that doesn't fit the vaginal opening, etc. Right? And so it's not going to happen. In gametic isolation, basically you have the gametes won't recognize one another. For postzygotic barriers, you have these after fertilization event to happen. We had a lively discussion during class about this, but in some cases you have reduced hybrid viability, and that the hybrid are born, but they're really weak and they tend to die, and they for the most part won't reproduce. You have reproduced hybrid fertility, where the organism may be very healthy, but it just doesn't reproduce. And you have what's called a hybrid breakdown, where maybe the first couple of generations it's healthy, but then over time it tends to kind of break down. And so the two types of speciation events that we have here, and look at it, we have what's called allopatric speciation, where you have a population that somehow gets separated physically or something like that, or you have these barriers put up so that those individual populations are able to go off. In sympatric speciation, basically it's where you have a population that's able to break off, and so you have individuals in that population have mating. Hello. Again, when we're looking at this, it's similar to what the model is. We have we have a fast model, and then we have a gradual model, and basically what's more of a punctuated equilibrium. And evolution tends to move fast until it doesn't move fast, and then it moves slow. And so again, macroevolution is a cumulative effect of many speciation and extinction events. Speciation can be quick or long. It just depends on the mutations, and it depends on the selection that it has. All right, so that's basically looking at speciation. The next thing, the next slide that I want to look at is basically the history of life on Earth. And so I set this up. So what is the history of life on Earth? Or more importantly, what am I going to ask you about the history of life on Earth? And let me click on this slideshow, and that will go on this, and it's popping up. And so basically, a lot of times when we look at history of life on Earth, again, there's a finite number of species that are out there, but again, there's really too many to look at. And when we're looking at history of life on Earth, we're looking at these kind of macroevolution, these broad patterns of evolution that occur above the species level. And so again, some of the big things that we want to know about in this evolutionary process is the emergence of terrestrial vertebrates, the impact of mass extinctions that they had on life in the planet, and the origin of key adaptations, such as flight, and maybe even the ability to use oxygen, etc. Again, the conditions on the early Earth. So again, we had a point where there was no life on Earth. Again, we look at Mars, we think Mars is almost sterile. We take a look at something like Venus, it's sterile. We don't know until we look at it. We're looking for life, but basically life had to come from somewhere, which is what we call this abiotic life synthesis. And how this happens is that the organic molecules will form. There's been a number of experiments on there, but we can form organic molecules not using living systems. The joining of these two molecules into what we call the macromolecules, the packaging of these molecules in protocells, and then evolution working on those protocells until we get something to life. And so we may have had something that mechanistically kind of looked a little bit like life, but it wasn't life. But again, in evolution, what you have is these things getting better. So if we had a non-living protocell with all these materials in there, and it was able to kind of reproduce, as long as it was able to reproduce and produce some more, we can look at these mutations and we can get it better till it was living. Your book tends to use this thing that we call a kind of clock analogy. I think of it a little bit, when I do my washing machine, I have this little knob on there that I can do it. But basically, we're looking at roughly about 4.5 or 5 billion years, which is a really long time. And again, if 5 billion years was a football field, we human beings would be around there for only a few millimeters. That was it. So if we go back, and so what I want you to be able to do is to be able to kind of relate this story of what's life and what and relate what are the large macro evolutionary processes that were in there. And so again, we have the origin of the earth. We have the origin of prokaryotic organisms. Again, we have the origin of oxygen. Once we had oxygen, organisms were able to do a lot more biochemistry because again, before the presence of oxygen, it was you really didn't get a lot of energy out of these things. Roughly about 2.5 billion years ago, we see the development of the single-celled eukaryotes, and again, due to those symbiotic relationships that we've had. The multicellular eukaryotes started to occur roughly about 1.5 billion years ago. Animals only came about, fish and things like that, roughly about 0.75 billion years ago. The colonization of land, and again, land was kind of an empty space. There was a number of things that lived in the ocean. That happened 500 million years ago. And again, we human beings are a relatively recent addition on that. It doesn't mean that we are the pinnacle of evolution, but it's been a while until we could kind of get here. Again, when we're looking at these things, life, we see these major radiations that are occurring, and they tend to occur after things like massive die-offs. And so things happen on this planet. A volcano goes off and does something. A meteor hits the earth. And when we look at the fossil record, we could see at certain times we would have these these large deaths occur. And so the number of existent families, and then the extinction rate. And so we had these very high extinction rates. Roughly, we had about 5 of them, 1, 2, 3, 4, 5 of these extinction rates. However, a lot of times after those extinction rates, we would see actually diversity go up a lot afterwards, because again, those organisms that live there would inhibit the new life forms from growing. Is there a sixth extinction? Other way? A number of scientists think so. Many species are declining rapidly due to habitat loss, overharvesting, and other factors. Climate change may hasten declines. And there's data that suggests that a sixth human-caused mass extinction is likely to occur unless dramatic action is taken. And so again, we have some very serious things that are going to be going on relatively soon. And again, when we look at the geological record, it's here right now. In our lifetime, it may not be for a little while, but when we do that. The next thing that I want to talk about, is how can we get these evolutionary process? And you would think of something like as a point mutation, not necessarily being able to cause that much change. But, again, in the evolution of development, we see that in times like the Cambrian explosion, we saw lots, so we had lots of life around. We had lots of multicellular life around, but at a certain point, we saw this kind of really big increase in diversity. And basically what we think these are the developmental genes, the genes that are involved in switching, again, you going from an infant to an adult. There's all these genes that cause an organism, a multicellular organism, to kind of change what it does over time. And it's changes in these genes that can have a big effect. And so an example of that, we have these things that are called Hox genes. And, again, if on the left-hand side, we have a crustacean, relatively simple ancestor. This ancestor is made up of a number of these segments, and each segment would have a leg pair. And what we would see is that we have these Hox genes that actually cause the suppression of leg formation from being formed. And so this way you have, you can go from a crustacean to an insect that has 6 legs. A crustacean may have lots and lots of legs. And so what this allows to have happen is this kind of, these segments are able to develop into different things that can support the organism. Again, when we look at evolution, we're thinking of evolution as more of a kind of tinkerer. you know, in evolution, you can't think about this is the goal that I want to go to, and then we're going to develop this stuff to go that goal. What basically is randomly happens. You change a few things, and then evolution kind of tries those out. Natural selection tries those out, and things that survive continue to be there. Things that don't survive will tend to go away. Right? That's my 15 minutes of talking about evolution. And, you know, again, on Friday, the 28th, we will have an opportunity to talk about this a bit more. So thanks a lot for coming, and I will see you later.